To this. I'll start that again. Welcome everyone to this webinar training session for the Standing Oak Tree Fungus Survey. During this session, we're going to be covering how to download and install the app, how to use the light version and useful features of Survey 123, what the key differences are between the light version and the full version of the survey, how to observe trees and fungi, taking good images, some background on oaks and their importance, and also a quick look at some key oak fungi that you'll see while you're out recording. I'm Rich Wright. I'm a PhD student at Cardiff University, and my project that I'm looking into is fungal heart rot communities in British oak trees, and I'm supervised by Lynn Boddy, who's here in the background, keeping an eye on everything, and Hilary Rogers, both at Cardiff. And I'm funded by KES2 and supported by Action Oak, the Royal Forestry Society and Welsh Government. I also work part-time at Royal Botanic Gardens Kew on the Fungal Tree of Life projects. So that's enough about me. During this session, if you have any questions that you definitely want myself to answer at the end, you can add those into the question and answers panel at the bottom of the screen. And of course, please feel free to use the chat session, the chat window, during the session for any light questions. And Lynn and Izzy, who are both here, will field and answer any of those as we're going along. And feel free to obviously communicate with each other in there as we're going along as well. If you haven't already downloaded the app, you can follow the QR code on the right there or enter the website address to download. You might find it useful to have the app open during this session to familiarise yourself with it as we're going along. Okay, so I'll now get started. So oaks are the third most common British tree, ash probably being the most common that's out there. And there are 120 million oak tree individuals within the UK. They can live for many, many years, some of them thought to be over 1,000 years, like Offa's Oak, for example, at Windsor Great Park, that's thought to be 1,300 years old, so an incredibly long lifespan. The UK supports more than 49,000 ancient and veteran oaks, more than the whole of Europe combined. And this makes the UK an incredibly important global site for lots of the biodiversity that's associated with veteran and ancient oaks and some very rare species. So oaks support over 2,300 species according to recent research, including birds, bats, bryophytes, lichens, mammals, and a vast number of invertebrates. Around 320 of these species are endemic to oak, and a further 229 are rarely found on any other tree. The highest diversity is associated with veteran and ancient trees. So veteran trees are trees that are in some way affected by fungal decay in their lives. And ancient trees, so many oak trees over the age of about 150 are affected by fungi in some way. Veterans are generally those that you can see clearer effects of decay. And the deadwood itself supports over 700 species, a large number of those being invertebrates. Sadly, oak habitat is in decline globally, which within Europe is considered to be the result of a broad range of anthropogenic and environmental factors that in combination have greatly reduced the area of woodland and the quality of that which remains. British native oaks are also threatened by a growing number of recently discovered syndromes, including acute oak decline, chronic oak decline, and are also constantly challenged by the native root pathogens in Armillarian gymnopus and functional leaf loss caused by mildews near Erysipelas. All of these are compounded by changes in land use, decreasing habitat and nitrogen deposition. Our work is focused on oak heart rot and the species of fungi that are involved. Heart rot, simply put, is the decay of heartwood brought about by a fungus and often assisted by other organisms in later stages. Heartwood is the non-functional wood that is formed from sapwood as it senesces. As a tree ages, it creates a large central column of heartwood, which contains few, if any, living cells. This process can create many benefits for the host tree and also creates complex microhabitats that support the amazing biodiversity I mentioned earlier. We're looking at this community that exists of fungi that exists inside the tree in a number of different ways. Firstly, we're surveying in heartwood using heartwood cores. This is taking a 45 centimetre core from about 1.5 metres up a tree into a standing tree, which does little harm. We're very clean and careful with our methods, and that allows us to extract a core which we can then culture from and also do molecular work to identify the fungi that are present in the tree. This is one of the few methods that allows us to actually see inside that column of wood that makes up the trunk of a tree, which is obviously otherwise impossible for us to look into. Most data that's gathered on fungi that are on oak trees is from observational data, which is what we'll be talking about in a little while as well. There's a number of other of these sampling techniques that allow us to see what's happening inside. We also do dowel sampling, which allows us to sample wood from the interior of oak hollows, like the one that I'm sitting in at the moment, and see which fungi are in the later stages of decaying wood. And we also work with slice sampling, which is where a tree has potentially fallen in a storm or where it's been felled due to construction work or something similar. We then get slices out of that tree, which we're then able to randomly sample over the surface of, and again work out where the fungi are and which fungi are present. And this allows us to build a sort of three-dimensional map of fungi throughout the centre of the tree. We also look at interactions experiments, pitting different fungi that we know exist inside oak heartwood against each other, seeing how they compete, seeing what defense mechanisms and things that they have. And from this gives us a little bit of information, insight into how succession may play out in the tree. So this and a number of other things are part of our research, but the key one that I want to talk to you about today is observational surveys of fruit bodies and tree conditions through the Standing Oak Tree Fungus app. So this app and community science project has been developed on the ArcGIS 123 platform, which offers useful integration with mobile devices, including GPS location, taking photos straight into the app, and a number of other useful features. It operates on any Android or iOS device and is available for PC and Mac as well. It can also be used in any browser, although this lacks some of the user functionality. The project is aimed at anyone with a knowledge of trees and the fungi that inhabit them. We openly encourage anyone who can tell a chicken of the woods from a beefsteak to get involved. All we ask is that you are very careful in your identification before submitting a record. The survey itself is only looking at standing oak trees, whether they're dead or alive. We're not looking at trees that have fallen down. This is due to complex community changes that occur when the tree comes in contact with the soil. So this would almost be another entire project in its own right. So we're just looking at the standing volumes of trees. To create a record, there has to be a fungus present. So we're not after records of just interesting trees or trees that appear to have signs of decay. There has to be a fruit body present for a record to be submitted. And we're also generally only really looking for fungi that are occurring on the wood of the tree. We're not interested in fungi that are growing on the leaves or on the acorns. This again would be a whole project. We are of course interested in fungi that are growing in the soil 1 to 3 meters away from the base of the trunk, because this would include species that are root rotting and potentially some mycorrhizal species as well. Recording can be carried out in a number of different ways. It can be done entirely ad hoc while you're out on a walk, and you can just record what you bump into as and when you have the time. You could also be more structured about it and plan recording at a particular local site or work through sites in your local area. This could be done through single visits where you just turn up and record a number of trees across the site over an afternoon or throughout a whole day. Or it could be done as repeat visits at different times of year, possibly including the early summer midsummer and autumn and winter seasons to capture different species at different times of year. Laetiporus, for example, can come out from May all the way through till September, so it could be considered as an early summer species. Buglossoporus quercinus, the rare oak polypore, is a species that really only occurs between June and August, with older fruit bodies being observable in September and October. And in autumn, of course, the highest diversity of fungi would be fruiting from oak trees during that time. All of these different visits can actually be combined into a single record using the edit survey function that's available in the app. We'll look at that a little bit later on. So to start off with, what I want to show you is how to download and install the app. So this is our project website. You can find this at foreverfungi.co.uk/oak fungi survey. And that will bring you to this homepage. This is what it looks like on a mobile device. And if we scroll down, you can see just at the top of the page there's a download now button. Click that and it will roll us down to these 4 clickable download buttons here. The first one allows you to download the new light version of the app, which is the version that I'd recommend for beginners, but also for anyone who wants to do more full surveys. This is still a useful version to have available where there isn't many tree features to record, or you only want to record a single species of fungus. There's the original full record version of the app, which takes a little bit longer to fill out and has a bit more detail, but it does also give us a lot more data to work with and will be more helpful for us to be able to make statements about oaks and their fungi in the future. You can download a PDF version of the guidebook from here as well, and you can also get your unique collector code. This unique collector code is simply something that will be sent to your email address once you open this up and put in your email, and that will allow you to add that code onto your records. Which means that all of the publicly published data around this, your data will be anonymous, but we can recognize your data and acknowledge you for it in publications. And also, should you wish to reclaim your data at a later point for your own use, then we can use this to search our databases and compile your data for you. So just to install the light version for now, we click on download the light version of the app. Oops. And that will take us through to this survey 123 page. Now, this is the bit that's potentially just a tiny bit confusing. So just follow things in this order. When we get to this screen, if you don't have the survey 123 field app installed, which is the container for our survey, you need to download that first. So if you click on, get it on Google Play or on the App Store, oops, sorry about that, then that will open that up and you can install it as you would do any normal app. Then once it's installed, come back to this screen and then click on open the survey in the 123 field app. That will then download our survey into the survey 123 app for use. Exactly the same process. If you want to install both versions of our survey, both the light version and the full version, you can just come back to the website and select download the full record of the app, and then just click to download that into survey 123. So once we've downloaded the app and installed the survey, we can see this screen. This is the loading screen when you open the app for the first time. And what you want to do is ignore these things here. It's asking you to sign in or manage anything. Just continue without signing in. This then opens the home screen, which contains all of the different surveys that you have installed in the app. First off, we can see here we have the Oak Light version and then the Oak Full survey version here as well. So what we're going to do first of all is I'm going to walk you through just the light version of the app, and we'll do that now. So this is the home screen of the light version of the app. And you can see down the bottom here, we have 3 different key options. We have Collect, which opens a new survey. And this allows us to then start putting in new data. So this is what we'll be using out in the field. And we have 2 other folders down here that contain previous surveys. Our sent folder contains the ones that have actually been sent to the uploaded already, which we can come back to and edit, and our drafts, which are surveys that have been not completed as yet. And we'll come back to looking at both of these folders and their functionality, which is really useful in just a bit. So to start off with, we will start collecting. And if I just open this alongside our visuals here, we can sort of have a little look, see what's happening as we're going along. So to start off with, the first thing that I'd like to draw your attention to is we have these 5 different sections here, and you can open and close those by clicking on them, and they each contain different sections of questions. The ones with the red asterisks are the ones that have information that has to be filled in. So not all of the questions, but they contain questions that have to be filled in, and the others do not. If we look at just the Your Information section here, you can see that it just has these green bands, which indicates the start of a new question, and then the fields to fill them in. If you're new to the app and you want additional help as you're working your way through it, if you click the Help and Support tab at the bottom, you can see there's a few paragraphs of information here with some useful links. But most importantly, at the top, there's this toggle button here, which if you click Yes on, it will display extra information and hints above or underneath each of the questions as you go through, which can be quite useful if you're unsure as to what needs to go in. Once you've got familiar with the app, it's a good idea to just turn those off, because it makes the whole experience a little bit more streamlined and there isn't so much text to confuse you.
Can I interrupt a moment? There are a couple of things in the chat. One person asked, can you put the QR code up again? But there's a lot of people having trouble. They say they can't download surveys without signing in.
They don't need to sign in. So we'll come back to that if that's, all right, let's just, the surveys won't require signing in. There should just be an option underneath to continue without signing in, as we saw on the original page there. There's going to be a few of these sort of technical things that are going to come up, but just because we've got quite a bit of stuff to get through, I'll try and answer some things at the end, but with other things, it's going to be useful if you visit the project Facebook group, where I've already addressed a couple of those in the discussion section of that page. And also just referring to sort of the guidebook, which contains information about how to get around that as well. But yes, if you want the QR code back up, I will just stick that back up for a second. There you go. Just stepping through back into the app here and the Your Information section. There's only 3 questions in here. The observation number is already automatically filled, so you don't need to add anything in there. If you want your collections to be marked with your collector number, you can enter in there, but it's not a requirement. Otherwise, all of the submissions, if there's no collector in there, will be anonymous. And then this other section here, the date of observation, which is only required if you're adding a historical record. So the app allows you to add backdated records. So if you've taken, say, photos of a fungus on a tree last September and you happen to have measurements of the tree, you can add that. when we open a new survey, it automatically includes today's date. So if you know the date that those photos were taken on of a particular fungus on a tree, you can add that in there, and that overrides this current date. But that's the only time you'd ever need to use it. Otherwise, you don't need to add a date at all. It's already included when you submit your survey. The next section here is one of the really important ones. This is the location details, and this includes at the top here the GPS location, which if you're on your mobile device will automatically start being detected by the GPS sensor in your phone. You can see that it shows things on a little map here, and there's the actual coordinate system and things at the top. And at the end here, there's this little couple of this plus minus, and it says 15.7 meters there. That's the accuracy or the margin of error of the GPS at the moment. And ideally, it's great if we can get that below 5 meters, just so that the positions are quite accurate, especially if you're working in the woodland where there's lots of trees present. To do that, you click this little circle with the crosshair on it, and it should start. Hopefully, I'm in a building, so it's not going to work particularly high accuracy. Out in woodlands, you should find it gets down to 3 or 4 meters without any problems. If you click that a few times, it will keep updating and hopefully slowly sort of whittle in. Leave it every sort of 10 seconds or so, and it will come down. But it's not going to get any better for me because I'm in a building right now. If you're unconvinced by the accuracy, or if you're adding a historical record, you can actually open up this map view and scroll around until you find the location that you want to put it into. You can use the search bar at the top here to search by postcode or by location. And then you can, of course, in the same way as you would do on Google Maps or something similar, zoom into the aerial view and perhaps select the exact tree that you wanted to add it to. Press the tick button, and that then secures the GPS location at that site. The next section we have here is the habitats, the broad habitats that the tree occurs in, which includes woodland, parkland and scattered trees, which is the type of habitat you might find on older estates, including woodland pasture, where there are scattered trees and grazing that occurs underneath it, so sort of mixture of larger trees and grassland. Lone standing field trees, which are common in agricultural settings, whether trees in a hedgerow, whether that's a narrow hedgerow or a wider one, whether it's in an urban situation like a street tree, or in a garden or a park, or by a river, brook or stream. You can, of course, select more than one of these answers. So it may be that you have a woodland tree, for example, that's by a river, brook or stream. The tree that we're going to talk about, we're going to say was in an urban situation, is on a road. And then there's a box here for additional habitat information as well. So into that box, we could add something like trees, roots covered in tarmac, which is a common thing along the streets around here. And maybe useful bit of information. So you don't have to include anything in here, but if you think there's something relevant about habitat, then please do include it. And we then move on to this next section, which is information about the oak tree. So in this section, we would like to know which species of oak it is that you're looking at. There's Quercus robur, the peduncular oak, and Quercus petraea, the sessile oak, as the two key common British species. There is, of course, to make things a little bit more complicated, the hybrid between these two. Identifying oaks, but even telling the difference between these two isn't necessarily an easy thing, and it certainly takes a little bit of reading and a bit of experience to be able to do that with confidence. So if you're not sure, please just tick "I'm not sure" which oak species it is. You can see these squares next to the labels for each of these choices. If you click on one of those, that actually opens up this little window with the images here to help you with identification. So you can see that Quercus robur has this slightly more dishevelled, slightly more squat sort of stature, tangled branches, the pedunculate acorns there, so acorns and a little stem, and fairly coarse bark. Whereas in Quercus petraea, we have the sessile acorns, they haven't got a stem, and it's generally a more taller and less sort of like messy looking tree with longer, wider spaced bark fissures. If you can identify the oak tree and you think that it's a non-native, you can select it from a number of species here, including the Quercus robur petraea hybrid. The next thing we'd like to know is whether the tree is dead or alive. And this is something that obviously can only really be told with any certainty in the spring and summer months when there are leaves produced. So if a tree has got leaves, no matter how few from a branch, then we would tick that it is alive. And if a tree late in the spring has still not produced any leaves, then we can fairly confidently say it's dead. And of course, if it's in standing but in quite obvious late stages of decay, even in the winter, we can be confident that it is dead. So the next bit is possibly one of the more tricky things, which is taking measurements. And this ideally should be done with a tape measure. Either a little tailoring tape measure with a couple of pins to help you pin it into the tree as you're working around, if you're working on your own, or using a tree measuring tape or surveyor's tape, which will obviously have greater length and is a little bit easier to use. If you're using a tape to measure, then you're measuring the circumference and you select that box there, and we'll be measuring always in centimetres and always at 1.5 metres up from ground level, which is dBH in some circles. dBH varies between 1.3 and 1.5. For our survey, we're doing everything at 1.5 metres above ground level, and measuring the circumference. That's what you then enter in there. If you're estimating the diameter, if you're estimating you don't have a tape, then you can use measurements. If you work out at home what the measurements of your hands are, and then your hand to your elbow, and then your hand to your shoulder, and then perhaps your whole arm span, you can use those to sort of like break down and roughly estimate the diameter. Of course, if you have a walking stick or something similar, or something else that you carry with you that you know the length of, you can also use that. But just to be entirely clear, this is for estimating the diameter. So if you're standing in front of the tree, this is the extents that you can see on the left and the right, not the circumference all of the way round. And that would be recorded also in centimetres and at 1.5 metres. There's more information on this in the guidebook and on the website. And there's another little bit of space there if you want to add any other information about the tree. So I'm just going to add in here that there are all lower branches cut off. We're talking about an urban tree, and in this, they often trim off the lower branches to stop them scraping a bus or a car or whatever else that's going by. And we then want to have a look at the photo section. This is adding photos of the tree. And we'll come on to looking at how to take good photos of trees and fungi at a later stage. But to do this from within the app, you can either click the camera, which will open your camera and allow you to take photos that come straight into the app, or you can click on the folder, which will open the files on your phone and allow you to select an image from there to upload. This is actually probably the preferable method if you want high quality photos, because the native camera app in your phone is likely to produce better quality and will allow you access to zoom features and various other things that might be handy. But the camera option is just nice and quick and easy as well. Let's quickly show you how that works. Take a photo from in here and There you go. That then pops into the app, and you can see now that both of the icons have a plus next to them. That allows you to add more photos either from the camera or from a folder again, and you can also delete photos as you're going along. Ideally, when we're taking photos of the tree, we want between 1 and 5 photos maximum of the tree, trying to capture details that we'll look at in just a moment. So that's all the information for the tree that we need. And then the next bit is information about the fungi on the tree, the final section. And this has 2 different methods in which it can be done. There's the simple mode, which gives us this gallery view, which allows us to select from these 20 different species, all with images to help us with identification. So we've got Laetoporus there, we've got Dahlia quercina, a nice view of the lovely maize gills. And so we could select one of these. So we found Fistulina, nice common one on oaks. Or alternatively, we can use expert mode. So if the fungus that we are observing, we know the identity of, isn't in this selection grid, we can select expert mode and we can add the scientific name into this field here. Now this is populated with a load of names already. So what I was observing was Ganoderma australe on our street tree. If the species that you're observing does not occur in this drop-down list, you can add its name in here. Just make sure it's been spelt correctly, please. We can then add photos of the fungus in exactly the same way as we did for the tree. And we'll talk more in a moment about taking photos of fungi and taking helpful photos. The next bit here is then how many points on the tree is the species fruiting from. So this is another one of the questions that's perhaps a little bit, has a little bit of grey area. And I'll just open up my slide that's going to help out with this. So what we're looking for is how many points on the tree that the species is fruiting from. So if we look at this tree on the left here, it has lots of pseudonotus fruit bodies growing from it. In fact, it had 75 fruit bodies growing from it all the way around, well, the most I've ever seen. But quite a few of those were in conjoined clusters and some of them were separate. We're interested in how many separate points there were. So in this case, it was actually 43 separate points that they were growing from. This is, say, particularly important if we were looking at, say, something like one of the armillaria, one of the honey fungus growing from the base of a tree, where we may have a single growth point. That growth point may have 50 individual stems and caps attached to it. That would just be recorded as one single growing point. If there's another cluster along that's also got another 50 caps on it, then that would then count as 2 growing points. I hope that makes some sense. There's more information on that in the guidebook as well. We also want to know whereabouts in one of these 5 areas that the fruit bodies you've observed are fruiting from. So in the case of our pseudonotus on the left here, we can see that it's growing definitely in the butt or root area below 50 centimeters. So we'll tick that one. And it's also growing in the main trunk area before any branching. So we'll tick that one. But it doesn't appear to be occurring any higher up the tree. So that's where we leave it at that. In the case of our Fistulina that we can see on the right here, it's growing in one location from the roots around the back of this tree. So we'd only include just butt or root area and a 1 in our fruiting points data there. We can then add any extra information about the fungus if we feel it's important. I currently don't feel there's anything else that needs to be added here. And at that point, we're all done. So to finish, what we can do is we press the tick button, and then that will automatically submit the record to our database. Or alternatively, what we can do is if we want to save this in drafts, so If you haven't got all of the data in, or you want to just make a quick record in the field and then add photos later, we can save to drafts. But also, if you're out in the field and you don't have mobile data, or you have to pay for your mobile data, it's better to save to drafts and then upload when you get home to Wi-Fi or the internet. So to do that, we press the X button and we click Save in Drafts, and it's then stored in our drafts folder. If we open up the drafts folder, we can see that there's 3 different records that I have in here. And actually, it'll start to tell us where each of those are. So these arrows tell us how far away each of those records are. So the one that we were just making, it's telling me is 0.7 meters away from where I am. And there's a couple of other ones earlier on here that are 100 feet behind me. I can also observe where these drafts are on this map view as well, which is very handy if you're returning to add more data in either the drafts or the sent items. So to re-edit a draft and to finally send it off, we just click on it to open it here. That loads the survey. We can add in any of the extra information or photos that we need to do, and again then just hit the tick button to send that to the database. Exactly the same thing happens in the sent folder. This is really useful, especially if you're using the full version of the survey and you want to come back at different times of year and potentially add in more information or more fungal records to different trees. You can actually find your position. This will have your GPS position on the map as well if it's close to where you are, so you can find trees easily by walking between them and then adding data to them in that way. So next up, I want to show you some examples of how to take good photos of trees. So I've only got really sort of like 1 to 5 photos that are possible to be taken. We don't want to have loads of photos of a tree uploaded, as it will make it complicated for us to sift through. So ideally, it's good to get a photograph of the tree at a distance, if that's possible. Obviously, in a woodland, that's much more difficult. So you can get an idea of its stature and just sort of overall features. But also we want photographs of the close-up of the trunk and up into the branches, and photos from an angle that are going to show any of the decay, wounds, or entry points for that tree. Taking photos of fungi is also really important, and we kind of want to get photos that give us as much information as possible, because we're going to need to go through and check the identity of each of the records, or records that are questionable at least. So this is a good thing if any of you aren't aware of this already, but if you're asking questions on, say, the BMS Facebook group or something similar about a fungus you want some help with identifying, then it's really useful to take a photograph that isn't just top down of the caps or top down on top of the bracket, and that you include the underside and the stem of the fruit body as well. So in this photo here of spindle shanks, you can see that I've picked a few specimens out of the cluster and I've laid them down next to it, so you can clearly see the gills, the attachment of the gills to the stem, and we can see the stem and the stipe base as well. That gives us plenty of information to make a good identification. And the same thing's been done with this foliota at the base here. If you're taking a photograph of a particularly small species, then do try and use the zoom feature on your camera. Or if you have a macro, then that's absolutely ideal, just so that we're not getting photos of little tiny white specks with the identifications. And of course, the most common group that you're likely to be taking photos of is the brackets, which would be the most common things occurring on oak trees. And for those, it's nearly impossible, I found, to get all the information that we need for a positive ID from a single photo. So take one from above, so you've got a nice clear shot of the bracket, and then one from underneath, so you can see the pore layer or tube layer or the gills or whatever else is underneath there. So now we'll have a quick little look at some of the key differences between the light version and the full version of the survey. Just quickly, so here you go. I've just come back out to just come out to the main page for the app, and then opened the full version. And from here, if I open collect, opens a new survey. Now, this survey takes a little bit longer, but delivers much richer data, which requires some knowledge of tree features and fungi. So it's laid out pretty much exactly the same. We have GPS location, everything else. When it comes to the habitats, this is the first key difference. There are a greater selection of habitats here, and there's also sort of like they can be combined more easily. So for example, we could have a broadleaf woodland which has a river running through it. We can have a grassland which is unimproved. We can have a number of different combinations, and all of those should be hopefully relatively self-explanatory. You can add again additional habitat information. Information for the oak tree have the same things as before, but we can now add in, if you know the age of a tree, and only add this if you do know the age of a tree, either through personal experience or through records of its planting, you can add the age of the tree in there. And you can also add in how the tree has been managed, whether that's through coppicing, pollarding, or monolithing. Do be careful, because lots of trees do sort of self-pollard as they get older. So we really want to only know if this has been something that's been done through humans. And again, there's little illustrations to help out with each of those, and more information in the guidebook. Measuring is exactly the same. And then when we're looking at the tree, in this survey, we want more details of the decay, wounds, and entry points. So whereabouts they occur in the tree overall, whether there's multiple areas that they occur in, you can tick and select a load of these. But when it comes to then making observations, there's a number of different features that we're interested in. And there's photos included for each of these to help out. So maybe you've observed woodpecker holes on the tree. We could tick those, and we could say that they occurred on the main trunk. Perhaps they occurred on the main trunk, and they also appear to appear in the lowest main branches as well. Perhaps the tree had buttress roots, these big roots that are sticking out at 45 degrees, so we can include those, and they'd of course be included in the butt or root area. And you can keep adding features as you're going around. So healed over wounds, lots of trees have these. That's a useful thing to include. Take your time making your observations, and then include as much information as possible. Adding photos of the trees is exactly the same. And then the next differences are just in adding information about the fungi on the tree. And in this case, we can add more than one fungus observation. So to do that, we can select how many we want to add, 1, 2, or 3, and then it adds in a separate little group for each of those to be completed. So again, it works in the same way. You can either work in simple mode or in expert mode. Don't switch between these two modes once you've started information. So if you know you want to add a species that isn't in simple mode, enter all of the information in expert mode, because if you don't, unfortunately, if you switch modes once you're moving along, it will delete the information that you've put in previously. If you check the help file for more information on that. So we want to know a little bit more information in here again. Where's the fungus growing on the tree, but also which part of the tree is the fungus growing on, whether it's on bark, whether it's on exposed wood, in a hollow, or on dead wood, or on soil around the tree. And that is the only key differences. So again, once we've finished, we press the tick button, or we can close it here and save in drafts. So now going to look a little bit about just how to make observations. When we arrive at a tree, it's probably a good idea to, instead of just opening the app up straight away, just to have a look around at it. Look at the roots, look at the trunk, look up to the principal branch union and higher branches, and have a walk all the way around. Look in small holes and inside of the hollows, and look for any signs of decay and entry points. And typically, especially in the case of things like fistulina, it's really common for there to be fruit bodies tucked away inside a little hollow or hidden underneath a burr. So doing this walk around will really give you an idea of what's present before you open the app and start making records. And as for where to find trees, you can visit your local woods and your parks. You can just find them when you're out and about on walks. But if you're interested in finding trees that are particularly notable, veterans or ancient, then you can use the Ancient Tree Inventory, which is a wonderful mapping service provided by the Woodland Trust and added to by lots of volunteers, that has records of many different trees. You can filter by species to oaks or particular species of oaks, and then use the map to find those in your area, and then go and visit and make records in that way. So we're now just going to have a quick little look at some of the fungi associated with oak trees, and then from there we'll then get on to hopefully answering some of your questions. So there are over 3,000 species associated with oak on the Fungal Records database of Britain and Ireland. Not all of these are specific to oak. There are some issues with these records in that associated organisms have just been labelled, and it's actually a tree that's just nearby. But at least 1,700 of these are associated with the wood. And that's 790 species of Ascomycete, 900 Basidiomycetes, and 15 in Zygomycota. And it's likely there's more to be added to that list as yet. So just going to have a quick look at some of these really key common species. Here we have Fistulina hepatica, the beefsteak fungus. This is by far the most common species that we've been seeing on oaks so far. And it's a fleshy annual bracket that's very distinctive, has this bright dark red to brown tones, and really does look quite alarmingly like an organ, like a liver or kidney. And it has a distinctive red guttation or red juice that can be seen if you squeeze the cut fruit bodies, and it's also produced directly on the mycelium as well, another key identification feature. It's very common on oaks in the UK, but is red listed in other areas of Europe, Scandinavia and India. And it also can sometimes occur as this interesting anamorphic form, Confistulina, that you can see on the bottom right here. It generally occurs in hot regions and certainly seems to be quite prominent around London. Another common species that crops up on oak is chicken of the woods, a very well-known fungus, this bright yellow and orange bracket that initially look a little bit like insulation foam in their young stages. It produces a brown rot in oak trees similar to Fistulina, but can also produce these large sheet-like mycelial plates that form through radial cracks in the wood. And that can be quite distinctive. If you see a large cracked oak branch or perhaps a cracked trunk, it's worth examining the wood around the crack area to see if you can spot plates that look like these. That's usually indicative of Laetiporus being present. There's actually around 19 different species of chicken of the woods now recognised globally. There's been a lot of taxonomic work exploring this group lately, and we're particularly interested to know if there are more than one species actually within the UK. So we're keeping an eye out for species that may look interesting like this, bright orange on the top and with a bright white pore layer, which may turn out to be Laetiporus cincinnatus. So if you do spot an interesting Laetiporus, please do get in touch. And then another really key species, the rare oak polypore, Buglossoporus quercinus, which is another fleshy annual bracket. It has a white flesh with a yellow to rich brown colouring on the pileus surface, which can be smooth or felty, and it bruises pink or brown when it's handled. It's one of only 4 protected species in the UK and is considered globally rare. And the UK is likely a stronghold, with Windsor Park probably being one of its focuses. It's only really found fruiting on trees older than 500 years, often from wounds or fallen branches, but can also occasionally be found on smaller, younger trees that are dead standing and very dried out. Pseudonotus driedus, the oak bracket, is another common species that can be found, especially in the west of the country. It's a very sort of like a lumpy looking bracket that produces red droplets of guttation, which is a key identification feature. Ganoderma species are perhaps some of the ones that are most easy to see all year round. There's a number of these that occur on oak trees, spursum and applanatum, lustum being rarer, as well as resination also not being so common. These have a white pore surface and can produce billions and billions of spores, creating these cocoa powder-like dustings of spores underneath the brackets, which are useful for identification. And then finally, we have Graphola frondosa, hen of the woods, a much loved species again, which can commonly be found growing from the base of oak trees, especially out in the west again, and is quite an easy species to identify. Oh, sorry, no, one more. There's the giant pippin polypore, Ripulus giganteus, which has a lot of similarities with Laetiporus, but it'll only really ever be seen growing from the base of the tree or from further away in the soil, growing from buried roots. Has a similar coloration but less undulating brackets, and as it ages or as it's bruised, it blackens. So its other common name is the blackening polypore. So that concludes this part of the presentation. There's more information on using the app and identification of trees and fungi on the project website, and also useful links on the resources page. I'd just like to, at this stage, to say a big thanks to our funders and also to Lynn and the brilliant team at Cardiff for making all of this possible. And I'd like to thank all of you for attending and also for submitting your records. We'll now work through some of your questions from the chat. Hi, Lynn, have you spotted any good questions in there so far?
Izzy has provided a current list of questions, I think.
Excellent.
And she's done a great job of answering questions in the chat as we've been going along.
Super. So I can just see one question in the question and answer section at the moment. Will the collected data maps be shared with participants afterwards? Yes. So once this is all actually the data's been checked and everything else, we will actually probably publish this to the website as a live map. So that will be something that will be coming along later on. And there is a hope, if we can manage to get funding and the agreement of the university, that this may be something that could be continued into the future beyond summer 2023, and in which case that would be a really useful thing to have up there to encourage people so they can see what's happening and what's already been recorded in their area. So yeah, thanks for that question, Guy. Whereabouts can I see these other questions, Izzy?
So there's 2 questions in the answered section, or I've summarized them in the main chat.
Nice. Excellent. Okay, so there's a question in there about the definitions of the age brackets: notable, veteran, and ancient. So I think Notable trees are trees that I think have just literally been things that are of interest, that have been planted perhaps by somebody important, or have got some local relevance to them. Veteran trees can be trees really of any age, as far as I'm aware, that have been affected by fungi, so hollowing or branch loss, or something that's really going to start to create the important microhabitats. And ancient trees, I believe, are going to fall into the category of over sort of 600 years or more. But I'm sure I could probably be corrected on that one. And the location data is, so the location data, well, we need to look into this, and if we're going to blur location data for edible species like Grifola frondosa and Laetoporus sulphureus, and I can completely understand your concerns with that and the data being used for foragers to go out and collect. When we've done previous things like this, then the data for edibles that has been blurred unless the data is being handed on to researchers or to record centres. So yes, we'll be careful with that. Great. Was there any other questions that you're aware of, Izzy or Lynn?
So somebody asked if they can add to their observation after they've submitted.
Yes, absolutely you can. So in the sent folder, you can revisit any of your records again. So I'll just show you that on the app screen now. So if I enter the sent folder and I go to this record here, you'll only be able to see your own records, by the way. This is only the ones that you've put into the app, not everything that's been uploaded to the survey. You can then either just view that, or you can click edit and resend here, which will allow you to add and remove any information or photos that you like, and then resend that from there. That's a really useful feature if you're going back to the same tree and recording throughout the year. Great. Any other questions that you can think of, Izzy?
Not that I'm aware of, but if anyone has any, I think they could put their hand up. Is that how that works?
I think in this webinar view, I don't think that is possible.
I did see if you go onto the list, we can give people permission to talk. So you can click allow to talk.
Okie dokie. Yes, and we've got Karen Corbell. Excellent. Yes, okay. Well, let's give that a spin. Karen, if you've got a question, we'll.
Oh, hi. Yes, I just wondered if the survey has start and finish dates.
Yes, so currently we'll be collecting data right up until the end of the summer in 2023, which is the end of my PhD. So I'll be able to keep including data up until that point. That's when my write-up's due. But I'm hoping that we can get it agreed that this will be something that will continue alongside our other research as well. So hopefully it will continue in the future. But for now, that's the finish date.
Thank you.
Brilliant, thanks. Super. Well, I can't see any other questions, so I'd just like to say thank you very much to everyone for attending. I hope that's been useful, and please do visit on the Facebook group for the app or get in touch. And yeah, good luck with recording, and thank you so much for your time and contributions. All the best.